This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Okay. All right, welcome everybody, and I'd just like to acknowledge the traditional owners on the land in which we're meeting and pay my respects to their elders past and present and elders from other communities who may be here today, and to acknowledge that sovereignty has never been ceded. And it gives me great pleasure today to welcome Sasha to present her PhD confirmation presentation on her project that she's working on, which I'll get her to introduce, with reptiles, parasites and pathogens in people. And Sasha I met 2001 or so, quite a long time ago, but she's lived many lives in zoo vetting and exotic animal world, knows basically. I'm watching her set this survey up, and it seems like she's contacting just friends and colleagues from many, many years, because you seem to know everyone across the sphere. So Sasha has worked as a zoo vet. She's worked at UVet for over 10 years in the clinic and teaching in the DVM, and many other hats, and worked at Lord Smith and as mostly with a focus on exotics and unusual pets. But yeah, also many different hats, and also key roles in our ethics committees over many years. So many of you would know Sasha really well. So yeah, I'll leave it to you, Sasha, if you want to take it away, and we'll please feel free, everyone, to put questions into the chat, and we'll just come around and try and make sure we address everything at the end and get round everyone. Okay, thanks, Sasha. Take it away.
Thank you very much, Simon. And I think I've got most of my supervisory team and some of my collaborators here watching as well. So hopefully it all makes sense as it comes together. So I'm aiming to investigate blood-borne microbes and their vectors in terrestrial lizards and snakes in Australia. As Simon said, I'm a clinical vet by background. I usually work with zoo animals, wildlife and unusual pets, and teach vet students at the University of Melbourne. But here I am doing a PhD and loving it so far. I would also like to show my respect and acknowledge the traditional custodians of the lands and of elders past and present on which this meeting takes place, which for me is the Djadja This is the plan for the seminar today. I'll introduce you to the topic and give you the definitions, explain what vector-borne diseases are and their general life cycles. We'll identify the gaps in bacterial vector-borne disease research, then look at the aims of the study that we've developed and the thesis chapters that we plan to put into place. I'll explain the study design and then go through the research schedule of what's been done so far, who our collaborators are and where, and the thesis timeline. There's a story of rickettsiosis, reptiles and mites, and that's me. In 2020, when the COVID pandemic hit, I became severely ill and was hospitalised with a persistent high fever and signs of meningitis, pneumonia and sepsis. I can't count the number of times I had my nose swabbed for COVID, but I persistently tested negative. Eventually, I was diagnosed with a rickettsial spotted fever. We didn't find out where it came from, but it was most intriguing. I kept being asked if I'd been bitten by a tick. The only parasites that I knew of that had bitten me within the 2-week incubation period was mites from a blue-tongued lizard. And that led me down the rabbit hole of rickettsial fascination and starting this PhD, investigating whether reptile mites are an undiscovered vector for rickettsia, amongst other possible bacteria. So vector-borne diseases. A disease vector is a living agent that carries and transmits an infectious pathogen to another living organism. Examples: this table displays the broad groups of vectors, hosts of vectors and pathogens documented in the literature. The mosquito-vectored viruses and the mammalian tick-vectored bacteria receive the most attention, alongside fleas and biting flies, but ectoparasitic mites can also act as vectors for bacterial pathogens and possibly viruses. Mites are poorly represented in the documented research on vector-borne diseases, but the knowledge is expanding. We know that the biting larvae of chigga mites infecting rodents, Leptotrumbidium species, transmit Orientia species responsible for scrub typhus in humans, and house mouse mites, Liponisoides sanguineus, transmit rickettsial pox caused by Rickettsia acari. More recently, spintonicid mites of bats have been implicated as vectors for Bartonella. So is my personal interest in reptile mites and a potential association with rickettsia warranted? In Australia, native reptiles and their tick, Bothropoda and Hydrosauri, act as reservoir hosts and vectors respectively for Rickettsia honii, the etiologic agent of Flinders Island spotted fever. But to date, the role of hematophagic and lymphophagic mites of reptiles, of reptile mites as vectors of such bacterial zoonoses have remained largely unexplored, despite their ability to vector potential pathogens to the host on which they feed, be that a reptile or a person. It is possible for trumbiculid mites to vector Rickettsia, so Orientia, but is it present in chiggers, in the reptile chiggers? Do other families of mites in other species carry rickettsia, or are the bacteria of potential vector-borne disease significance in humans? What does the literature on mites say? Well, in a collection of studies that have gone back over the trumbiculid mites and the Monocydidae and Mesostigmata in general, the mites on other taxa are shown to vector Rickettsia, Orientia, and Bartonella species. And as this table shows, the chigger mites, which are the trumbiculidae, collected from mammals, a few from birds, and some from reptiles, have been found to carry several other bacteria that are known to cause vector-borne diseases. Some of the other types of mites, Mesostigmata, have also been shown to carry some of those bacteria. And those Mesostigmata also are suspected to be potential vectors because they can survive prolonged fasting. They're everywhere, and they can, because of this resistance to the long periods of fasting, it makes them good vectors for dissemination and conservation of pathogens, and also suitable agents for maintaining an area of endemic disease, as Morrow in 2005 was already pinpointing. But not a lot more work has been done really since then. Reptile mites are rarely studied. You can see the numbers in there. In the Moniusco's study on the chigger mites and what they can carry, they looked at the 28 genera of chiggers, 100 species that have been shown to be associated with 31 different bacterial taxa, and not much of that's been explored in Australia. It doesn't mean that they're vectors necessarily, but they are associated with these bacteria. Let's bring that lens back out again and look at which bacteria we might find in ectoparasites generally in Australia. We've got mosquitoes, ticks, fleas, and mammalian mites, which are known to vector bacterial diseases in humans and animals in Australia. We have ehrlichiosis in dogs from ticks, anaplasmosis in cattle from ticks, Coxiella in humans, which is mostly by inhalation, but can potentially be transmitted by ticks, scrub typhus group rickettsiosis in humans, so Orientia from chiggers, spotted fever group rickettsiosis in humans from fleas on dogs, and ticks on shingleback lizards. If we have a look at the distribution, particularly of the Rickettsiosis in humans in Australia, the Orientia are up north, but haven't been really explored further in different places in Australia. We'll look at another map on the different places that have looked for scrub fever in a moment. The different types of Rickettsia are found all around Australia, as you can see. So, looking at mite-borne pathogen studies in Australia, this particular map comes from a review by Elliott in 2019, looking at scrub typhus ecology, so Orientia in vectors and hosts. Most of those studies have been done outside of Australia, and only very few have actually looked at it within Australia. Mites also have the potential to carry and possibly transmit Anaplasma, Bartonella, Borrelia, Coxiella, Francocella, and Rickettsia species if they are present. So what are the candidates for reptile-associated bacterial vector-borne diseases? We'll look at the bacteria, the reptiles, the mites, and the humans at risk of zoonoses. Rickettsial life cycle. So specifically looking at rickettsiosis, the rickettsial life cycle involves a vertebrate reservoir, such as a rodent, a dog, or a lizard. Sometimes there's an arthropod reservoir, and it's the vertebrate merely acts as the host of the arthropod. The ectoparasites that live on the hosts are the vectors, and that might be a trombiculid mite, a flea, a tick. They feed on the blood of the hosts, circulating the bacteria from host to vector to parasite and back again. Rickettsia are also transmitted through each life stage of the vector, from adult to egg to larvae. And the cycle doesn't normally involve a human until we accidentally invade the environment where the parasite vector is. Our reptile candidates, we're looking at both wild and captive ones. The only thing that has really been looked at reptile-wise is the sleepy lizard, Telequa rugosa, has been found to carry ticks positive for rickettsia in Australia. This lizard has also been found to carry chiggers, so native mites, trombiculid mites, and in some areas, mesospid matted mites, the Onatricus, Ophionisus natricus, which is an introduced mite. Blue-tongued skinks in Victoria have been found to carry Ophionisus natricus, and it's also been found on wild snakes. In captivity, Ophionisus natricus is a common problem of collections of snakes and lizards of many different taxa. So we propose looking at a broad range of different taxa of snakes and lizards, including skinks, monster lizards, pythons, and elapids, depending on which regions of Australia. We'll look at 5 different ecoregions across Australia, and we'll look at both wild and captive populations. Looking at the mite candidates as vectors of bacteria from reptiles, the trombiculid chigger mites are usually the native mites in Australia. Ophionisus natricus is the introduced one. Both of them are hermatophagous mites and not all that fussy about their host. They have free living stages, and they belong to families or genera that are known vectors elsewhere or in other vertebrates. They may feed from reptiles identified as reservoirs locally or in other geographic locations. And chiggers have been collected from reptiles, as we saw, have been found to harbour various different bacteria. Ophionisus natricus has been found in Brazil to be positive for one of the rickettsia, but nothing further was done at that stage on that study. My main suspect as a vector with the potential to transmit things throughout Australia and spread bacterial vector-borne diseases is Ophionisus natricus, which can leave its primary host to feed opportunistically upon other hosts of the same or different species, including humans. It's not fussy. It can cause a rash in humans. It's actually not all that visible on that slide there, but it is known to be zoonotic just purely from causing a rash point of view. It causes terrible disease in the reptile host itself. And it can also transmit, potentially, vector-borne diseases. It's known to mechanically transmit Eromonas between reptiles, and that's a fatal septicemia in reptiles. These mites are well qualified to be vectors. The human candidates, human accidental host candidates, we will look at occupational risks or hobby risks to arthropod vectors of bacteria. Professions and hobbies exposed to reptile-associated arthropod vectors might include ecologists and field naturalists, wildlife vets, wildlife park rangers and keepers outdoors, and then anyone who cares for reptiles that are in captivity would also potentially be at risk. Now let's look at the establishment of the research questions, the aims, hypothesis, and how that leads into the chapters for the thesis. Our aims are to assess the prevalence, diversity, distribution, and associated risk factors for vector-borne bacteria in terrestrial reptiles in Australia. We'll investigate the role of the reptile mites as potential vectors for blood-borne bacteria, determine the seroprevalence of and associated risk factors for spotted fever, typhus, and scrub root rickettsiosis in terrestrial reptile handlers in Australia. In reptile handlers, explore the presence of circulating DNA identified as being potentially zoonotic when we've looked at the blood and parasites in reptiles. So the hypotheses and chapters are displayed here. So the chapters are based on each of those hypotheses. The chapters we'll be looking at are, first chapter being the morphological identification of mites and ticks found on snakes and lizards in Australia, with notes on vectorship characteristics. We expect to find mainly chiggers on the wild lizards and Ophionicis on the captive, and possibly a mixture of the two crossing over. Chapter 2 will be bacteria found in mites and ticks, collected from snakes and lizards in Australia, including species for which the reptile host is a likely reservoir. And we expect a diversity of bacterial species will be found. Chapter 3, the prevalence, diversity and risk factors of blood-borne bacteria found in reptiles. Again, we expect there to be a diversity of bacterial species, and that will probably include Rickettsiaceae, amongst others. And finally, a cross-sectional study of the prevalence, diversity and risk factors for exposure to reptile-associated blood-borne bacteria in reptile handlers in Australia. Then the study designed to achieve the answers to these questions. My research is looking to characterise blood-borne infectious agents in common to reptiles, the ectoparasites, and the people exposed to them. For this, I need to collect blood and parasites from the reptiles, blood from the people, and get the people to complete a questionnaire to allow us to analyse the environmental and human-driven risks in relation to the prevalence. The prevalence study will compare findings across the major ecoregions of Australia based on the terrestrial ecoregions established by the World Wildlife Fund. And we'll break the populations down into wild and captive terrestrial snakes and lizards. So the main ecoregions we will look at, we'll leave out the montane areas in Victoria and New South Wales and the broadleaf tropical rainforests, just from a matter of time and small sample sizes available there. But we'll be looking at the arid region, Mediterranean forests and shrublands, temperate forests, temperate grasslands and savannas, and tropical grasslands, savannas and shrublands. For analysis, we will collect the samples shown here, and our sample sizes have been statistically predicted as shown here. We used this calculator, ShinyVet, which is based on EpiR package to calculate our sample sizes. We used a published prevalence of mites on reptiles as our estimated prevalence of 10% for the calculation, and assumed that reptiles living together, clustered together, would have very similar mite burdens and blood-borne bacteria, and that reptiles living separately in different locations would be very different from each other. Samples are grouped according to ecoregion of origin, captive or wild habitat, and snake or lizard taxa. Reptiles with and without parasites will be sampled. If we take 2 samples per taxa per wild or captive site in each ecoregion, then in total we need samples from 124 lizards and 124 snakes from each of the ecoregions identified to provide us with true prevalence with accuracy of 10% and 90% confidence. The statistical predictions were calculated considering the main aim of identifying the presence of and genetically characterising vector-borne bacteria in humans exposed to terrestrial reptiles in Australia. The human participants will be chosen based on their demonstrated exposure to reptiles. The prevalence of reptile parasite-associated blood-borne bacteria is expected to be lower in people than in the reptiles that they care for. A prevalence of 5% or less was presumed for these calculations. Taking blood from only 1 human per site, we need a total of 360 reptile carers for our study. The questionnaire on risk analysis was developed based on the causal associations we looked at using the DAG tool available here. The Qualtrics platform was used for questionnaire data capture. It includes exposure, the purpose of being exposed to the reptiles, the frequency, exposure, and the types of reptiles, biosecurity, the number and sources of reptiles that people are exposed to or to each other, and the hygiene routine, husbandry of captive reptiles, and then looking at the human health history and practices, including their illness history, personal protective practices, and hygiene routine. Using Qualtrics, we distributed the questions in those categories into the sections on the right here, and then we will use skip logic to be able to get the questionnaire participants to go through to the sections that are relevant to them, to both their profession or hobby and to the questions and answers they provide. Looking at the diagnostic techniques that we plan to use, we won't be using microscopy. It's notoriously insensitive and we often get false negatives. We're unlikely to be able to distinguish the different bacteria morphologically, and even heavy infections can be missed. We will use serology, but only on humans. There's no serological techniques, processes established for reptile serology in this situation. So we'll be looking for antibodies specific to rickettsia. That will give us a general idea on exposure to spotted fever groups, typhus groups, and scrub typhus groups, but not the specific species. Sometimes you get alternating high and low levels of antibodies and antigen. So the bacteria, when they are, for rickettsia, if they are suppressed by the antibodies, then you may get a negative PCR, and vice versa. If you've got high levels circulating in the bloodstream, then you actually usually means you've got low antibodies because they're not being suppressed. We'll use PCR to target rickettsia specifically because they are quite hard to find using the other technique which we're looking at, which is next generation sequencing, which we'll use more for the broad metabarcoding of looking for bacteria in general, not for specifics. So the difficulty with PCR, conventional PCR, is you really need to know what you're looking for before you go there, and there's not much information on what might be found in reptiles or mites, so we have to make assumptions. Next generation sequencing provides us with the ability to look at broad taxonomic groups and not specifics necessarily, so you can potentially find things that are novel, but it's not great at finding rickettsia. Laboratory analysis will take place in both Parkville and at the University, in Parkville at the University of Melbourne Veterinary Biosciences Laboratories and at the Australian Rickettsial Reference Laboratory in Geelong. Parasite species identification will be performed morphologically, and then we will look at molecular analysis of the blood samples and the parasite samples. The order in which we do that will be, first of all, we'll look at the human serology at ARL in Geelong, and NGS, so next generation sequencing, broadly for a whole lot of bacteria, on 10% of the reptile blood samples. We'll choose those that have been exposed to the ectoparasites for that 10%. The next step will be targeted PCR on the remainder of the reptile blood samples and on the mites and ticks, and sequencing of those positive results. Then finally, we'll look at the human whole blood molecular analysis and sequencing based on ectoparasite samplings findings. Greater than 5% of a species of mite or tick carrying a specific bacterium in high copy numbers supports vectorship. Bacteria specifically linked to a single species of mite or tick supports likelihood of transovarial or transstadial vectorship. High copy numbers of a specific bacteria in multiple individuals of the same species and region, so the reptiles, supports reservoir stasis. And then why are we doing this? The potential benefits include the identification of possible vector-borne disease agents and factors that may increase their spread within and between reptile colonies in both captivity and wild, and factors that may cause them to transmit to humans. We'll also identify geographic areas of risk for these vector-borne agents. And this provides a baseline on which to advise control procedures to limit or mitigate the risks of reptile-associated vector-borne bacterial diseases of both humans and wildlife. So the reptiles, we may find pathogenic agents that could affect reptiles in the process as well. Finally, let's have a look at the research schedule. So far, the research project, to begin with, I undertook training in epidemiology and the statistics in predicting the sample sizes. human phlebotomy accreditation, so I can collect blood from people and not just animals, genetic extraction training in the laboratory, and ethics, research, ethics, information training. The ethics applications have been completed, and both the human and the animal ethics and their associated documentation have been approved as of the start of January. We have submitted a broad application for a wide range of reptiles to the Victorian government, and we already have approval for the study on skinks. These three things will be used as a pro forma then to apply for permission collecting in other states and also to zoos and museums through their ethics committees. We have established collaborations with unusual pet vets and exotics clinics across Australia. We've established connection with the Victorian Herpetological Society. There's an expo coming up in a couple of weeks that will have about 5,000 people, where we will have a booth and we will begin collecting blood samples and looking for volunteers to have their reptiles sampled on another occasion and for people to begin doing the questionnaires. We'll look at the herpetological field sampling. We've made many connections through the Australian Society of Herpetologists from the conference last year and ongoing communications. And we've established connections with wildlife networks in all states and zoos and museums. We've presented the research already at 4 different conferences. So that's the plans for the research, obviously no results yet. And established a lot of in amongst the wildlife health communities and the Wildlife Health Forum and epidemiology within the University of Melbourne. This is our collaboration map, and our collaborators here, the stars are people associated with wildlife, and the others are people associated with captive reptiles. You can see they cover all ecoregions. By no means do they all look at reptiles only where they are based. They do care for reptiles further afield, whether that be in a scientific sense of research or a veterinary sense or a wildlife caring or reptile control point of view. This is my thesis timeline, and those that are enclosed boxes, filled boxes, are things that have been completed, and the open boxes are the plans for the next 3 years. When I say we, I'm talking about a research colleague who is going to be collecting samples with me and helping with all of the different stages around Australia with the collection of samples, collecting her own samples on antimicrobial resistance at the same time. That is Alice Aldora. So we'll be doing a lot of the sample collection together. I'd like to thank my supervisory team, which is Beck Traub, Simon Firestone, Lucas Huggins and John Stenos, and the funding that's provided by associations with all of those people. University of Melbourne, last year the Traub Lab, this year will be the Australian Reptile Reference Laboratory and the Cybex Foundation. Thank you very much.
Well, it was wonderful, Sasha. That you're relieved to get through that and very quick. So there's plenty of time for discussion and questions and comments from anyone out there that wants to ask things. I might start you off while everyone's collecting their thoughts and starting to pose some questions. But you presented sort of definition, a loose definition for what vectorship might be. And then also when we think about what things might constitute a reservoir, sometimes we think about more than just it being detected in this one species or the amounts. Do you want to talk about what might constitute a reservoir, in your opinion? Or is it a Beck's topic? You might do it.
Oh, goodness. I don't know. I could say, what do you mean by broadly when you were saying different species? And
yeah, so
I didn't really understand the question epidemiologically, sorry.
No, that's okay. What would you, like, if we just find things in a couple of different species, would you consider them a reservoir in themselves, or do you think more, what do you think about?
Okay, so if it's found in a lot of members of one species, and their parasites are also found to contain that bacteria, then it's more likely to be a reservoir. It's not just, it won't be a matter of we find it in one reptile that shows that it's a reservoir. It only says that it is possible that it might be able to be a reservoir. If we find, for instance, in skinks like the sleepy lizard, if we find it's positive for a particular type of rickettsia in its blood and in its parasites in multiple different regions of Australia, then that's more likely to be a reservoir.
Yeah. And I suppose the same goes for the vectorship. You don't want to stick to hard and fast definitions, do you? But yeah, you've got to think about what you, yeah.
Well, the vector can potentially be the reservoir too. Like some of the ticks have been shown to be, I think the ticks on Flinders Island, 100% of them were positive for the rickettsia, but the reptile itself has not necessarily been found to be positive for that particular rickettsia with those ticks. So, yeah, can't make assumptions.
Yeah. All right. Anyone else there want to put their hand up or ask a question in the chat? We're very happy to hear from you.
I had a question quickly for Sasha. Just going back, Sasha, to your origin story for this PhD with your experience getting sick at the start of the pandemic. Were you diagnosed with a rickettsiosis in the end, or I can't remember how it happened?
Well, I was from the human clinical perspective, the clinical disease and the positive serology diagnosed me with the spotted fever group rickettsia, but not able to say which one it was because no PCR was done, and unfortunately I had no nice little tick scab or tick to provide, and certainly the mites were well and truly gone, and the reptile that had the mites was treated, so there was no way of testing that. So yeah, so we didn't find out exactly what it was, but for a diagnosis, they usually require serology and clinical signs progression.
Yeah,
it wasn't fun.
No, it didn't sound it.
I plan not to get it again. I have absolutely no way I'm going to get another vector-borne disease despite studying them. The question in the chat.
Raises well, we'll wait for someone else. It raises a good comment for how you're going to put your findings out there, Sasha, because you're going to come across a lot of wildlife owners as you're doing this, and people interacting with wildlife, but you're going to find a lot of things of real interest to a lot of people.
Yeah. Mike, I can go along again, hopefully, to one of your conferences. Were you going to ask me something?
Yeah, I was going to ask you just how many samples of lizards and snakes and things are you getting? Because I think you had the numbers of the people that you're surveying and everything like that, but
so it was 1,240 altogether with all of the reptiles across Australia, wild and captive, snakes and lizards. So dividing that across the different 5 eco regions and across the taxa. So I can't do the calculation in my head on the stage right now. I think it ends up being about 60 lizards. So, for instance, 60 captive lizards from each ecoregion and 60 wild lizards from each ecoregion.
So you're trying to get anything that you can get? Is that the idea, or is it?
Yes, yeah. So it's limited by the size of the animal that I can get blood from.
Right.
So that will tend to be the larger skinks and monitor lizards. Probably not geckos. Will more likely being snakes wise, it will be pythons up north and the bigger elapids down south that I'll go out with the snake catchers to collect that and work with the captive herpers as well.
So just thinking about the numbers and things, because if you've got a very low prevalence in each species, so you've only got 10% or something like that, so you might not be definitive what you find, I suppose. Yeah,
that's true, and there'll be some things that will be statistically significant, and then breaking it down into smaller and smaller groups, it's not going to be statistically significant. It's just going to be an examination of what we find for the smaller breakdown of groups. Yeah, for sure. Yeah.
Because if you wanted to get, you'd have to get that number of each species or more or whatever. So yeah, I guess it's
for which I would need to be doing this for the rest of my life.
Yes, that's it.
We've got a question from Nick. Are you doing exotic reptiles from zoos as well, or just Australian natives? It'll be exotics too. It's because they will be all captive in the same area.
We'll rip around Alistair and Garrett in a second, because I think they've put their cameras on to us. I was just wondering, your numbers, and we've been asked this before, Sasha, your numbers seem really big, and Mike's outlined, even though they're big, the limitations of what we're trying to achieve. You might not even achieve it with those numbers. How are you going, and how do you feel about getting those sorts of numbers? Because you had a pretty good response when you first went out, haven't you?
Yeah, and so we went out, started collecting in December, and it's some of the collection we'll be doing ourselves, and others will be, for instance, the vets that we have engaged that work in different states. They will collect the samples for us, and obviously I'll need to maintain contact to maintain enthusiasm amongst all of the collaborators for collecting. If there's a researcher in Broome who is working on monitor lizards and already has permission for collection of blood and parasites, so they're able to collect some things for us when they go out again, depending on how the flood damage is after all of the floods up there in West Kimberley. And yeah, so I guess it's a combination of us collecting the samples and other people collecting the samples. Certainly we've had no trouble in garnering interest and starting to plan our schedule, but we'll see how it goes.
We'll move on to Alistair. Yep.
Thanks, Simon. Thanks, Sasha. I did try to put my hand up, but it kind of blends in with my background. I realized kind of not the most ideal color. You've somewhat asked my question for me, Simon. It was kind of around sort of, I guess, success to date that you'd had, and just to give you a bit of an idea of looking forward for some of those wild populations. Do you have any feeling, and apologies if you mentioned this already, feeling about scenarios where you might not be able to get the 60 animals in any of those eco systems that you talked about, particularly things like desert, where the animals might be present but sparse or hard to sort of come across? And is it just a case of going, well, that we tried and we couldn't get it?
Yeah, and look, I guess part of that is working with people who know the areas. And for instance, Garrett has just asked a question there: have I considered the period when we'll be doing the sampling, as some parasites may be very seasonal? And yes, indeed, that's going to be the case with most of the wild reptiles. Up north, they might have a longer active season, and especially when it's not the very arid areas, but even the arid areas actually still have quite a lot of life in them. So it will be more about doing the captive reptiles in those down seasons, Garrett, and then the wild reptiles in the busier seasons when the parasites are, well, both the reptile and the parasite are active.
Thanks, Sasha. It's a lot of work, so I think it'll be really excellent when it's all done.
It'll be good, and I guess we'll have to make changes as we go, depending on what we find, and that was very much the process of developing the study too. I guess is not just focusing on rickettsia, but more broadly on other bacteria. But if we find a new rickettsia, that might take us down a different pathway.
Yeah. Wouldn't be a PhD if you didn't have to make changes partway through.
It's just the life of an exotic vet. Hey, I'm used to that.
Garrett, I think, wants to add to that. Yeah, go for it.
Yeah, I'm just curious, with parasites like the chiggers, if you know their life cycle, you know which seasons to sample, but not all reptile chiggers have been studied. So how do you know when to go and find them? You could sample an area where they are present, but if you're there when they are no longer larvae, you're not going to find them on the reptiles, because only the larva is parasitic. The adults are free living.
Yeah, that is going to prove difficult, I guess, and we won't always find those. And speaking to the ecologists who I go out with, like you, about that and what they see, what they find anecdotally that's not necessarily in the literature, will be an important part of what I do, is listening and following the advice of the people who I'm sampling with.
Yeah, you've exhausted the question, Sasha, the discussion, which is well done. And yeah, what do you expect to find, Sasha, or what sort of excitement are you thinking of will come your way?
Oh, I think the fact of finding what's there in any of the parasites that we find with the mites, particularly. I mean, it is the mites that we focus on. The ticks is only if they're also co-feeding on the reptile. We want to compare what we find in each of them if they're both on the same reptile. I think it would be fascinating to find the same bacteria in multiple different geographic regions, ecoregions. I think that would be very interesting. I think finding things in the introduced reptile mite will be fascinating, but also slightly worrying, because that reptile mite escapes so easily and spreads so quickly, and easily becomes a pest. So it's a concern. It's a useful piece of information to find, but maybe worrying. But I feel certain that we'll find rickettsia there, but whether or not we find it in a new rickettsia or in the host as well as the vector, or evidence of that being transmitted to humans, I don't know. It's all interesting.
It's good unexplored space, and you're going to be happy with whatever you find, I think.
Yeah. Yep.
It will be interesting to see where we can cross over and speak to maybe the communicable disease colleagues in the departments of health once you've got some results, and see what they, or maybe through John Stenos and others as well, and just see what they're finding around the country. Yeah. John, you might already know or sit on that data. I'm not sure if you want to comment.
Sorry, I missed that question. What was it?
I'm just saying, once Sasha's got some results and some rickettsia and speciated a few things and got a bit of an understanding where some things are, will you have capacity to look at some of the health records, or is that something we go to the communicable disease department and the epidemiologists there and look across?
Yeah, look, those records would be limited to what we have in our lab. So yeah, it might be worth spreading a little bit wider catchment and going to the bigger authority.
Yeah, we can definitely approach who we have to when it comes to that.
Yeah. We're ready to roll, and we may roll down a different hill.
Well, I think the other side, because you've got Alice's work as well, parallel work looking at the bacteria in the fecal and in the oral clay swabs. There's Alice. Then we may even also find out some really interesting things about some of the salmonella and other zoonoses. That we know there's a few what they're called cryptic reservoirs. So we're just but they're blamed on various wildlife species that some of them might relate to reptiles for sure. No, Alice, did you want to say something else, or you just saying hello?
Yeah, just saying hello.
Offering support. Good one.
It will be. Right, well, it'll be interesting to see what we won't be able to speciate the chiggers. Probably. Well, I certainly won't be able to. They're quite hard, even for experts, to be able to speciate. So that's an aspect of the research too that potentially could get somebody else on board who wanted to know about the distribution of trombiculid mites on reptiles in Australia. So we will look for more people to join us.
Lots of spin-off questions, because I know just from Alice's early days in the lab trying to look at the bacteria you've been finding, and I think your experience before we even started this project with the microgroup was that the taxonomy and the regular biochem tests just don't really fit what we know. It's not really been explored enough to say, to sort of follow the usual keys and patterns. So I think everything you find is going to be informative. So it's an exciting space, Sasha.
It is. It is.
All right. Well, I think if everyone's quiet, we might leave it there, and just also well done, Sasha, on a great presentation today. You've overviewed what you're up to really well, and yeah, there's a lot of interest, and I hope this also spreads out. People now know to contact you about the study as well, so that might feed into getting a few more opportunities for you to sample. So yeah, thank you everyone for coming and your attention, and yeah, we'll just say well done to Sasha again. So,
Thank you very much, everyone.